Hi, everyone. My name is Tori Bauer-Pisani. I am a first-year graduate student at Southern, and I'm an animal technician with Yale University. So my paper is a negative feedback loop between microRNA 378 and NRF1 promotes the development of hepatosteatosis in mice treated with a high-fat diet. There's some background information. I have the terms identified in the next slide, so excuse the confusing acronyms for a second. NAFLD affects 20 to 45% of the Western population due to obesity. NAFLD carries a benign prognosis, but then leads to NASH and then cirrhosis with a risk of developing HCC. The pathogenesis of NAFLD has been studied extensively. However, precise mechanisms are still under investigation. Some of the factors hypothesized are insulin resistance, impaired FAO, and alterations in microRNA. The microRNA 378 that is specifically analyzed in this paper arose from interest in studies in the livers of dietary obese mice. The high-fat diet induced the expression of the microRNA 378 in the livers of mice, which microRNA 378 is embedded in the first intron of the PPARYCL gene, which encodes peroxisome proliferator activated receptor Y coactivator 1, a key regulator of mitochondrial biogenesis, thermogenesis, and glucose and fatty acid metabolism. The rate of microRNA 378 and hepatosteatosis and its dysregulation in fatty livers is not well described. Therefore, it is being used in this paper to study further. NRF1 is a potential target of microRNA 378. NRF1 functions as a transcription factor which activates the expression of some key metabolic genes regulating cellular growth and nuclear genes required for respiration, hemibiosynthesis, and mitochondrial DNA transcription and regulation. Replication, not regulation. Hepatocyte-specific knockout of NRF1 causes liver pathology similar to human NASH, and it's hypothesized beforehand that a negative feedback loop between microRNA 378 and NRF1 promotes hepatosteatosis. The study is designed to investigate the regulatory mechanism of increased microRNA 378 in fatty livers and elucidate how microRNA 378 promotes hepatic lipid accumulation as an established mouse model. So the terms identified within the paper, NAFLD, it's non-alcoholic fatty liver disease, NASH, non-alcoholic steatohepatitis, HCC, hepatocellular carcinoma, FAO, fatty acid oxidation, FFA, hepatic free fatty acid, HFD, high-fat diet, NRF1, nuclear respiratory factor 1, and SREBPs, steroregulatory element binding proteins. Some of the methods in this experiment include the establishment of dietary obese mice, 8-week-old wild-type mice with the strain C57BL6 were maintained in either a standard or high-fat diet for 8 weeks. After the time, the livers were collected for microRNA and gene expression analyses. Then moving on to preparation of mini-circle expression vectors for microRNA 378 and shRNA of NRF1. These mini circle vectors were transformed into a special host E. coli bacterial strain, and the verified shRNA of NRF1 was injected into the mini circle vectors, and the TTR promoter was used to ensure hepatic expression. And moving on to MCTTR, MIR 378 treatment of dietary obese mice. The male mice on the high-fat diet were kept on that again for 8 weeks and then divided into 2 groups. One treated with the MCTTR microRNA 378 and then another with a control. And those were injected weekly for 8 weeks via the tail vein injection. And at the same time, the mice were anesthetized and blood was collected by cardiac puncture. The livers were also harvested and immediately frozen in liquid nitrogen for gene expression and histological analyses. And then moving on to the generation of microRNA 378 ASO. and that was produced by an efficient cellular uptake of the oligonucleotide that carried a fully phosphorythioate modified backbone. Then there was an intravenous injection of microRNA 378 ASO. The ASO was resuspended in sodium chloride and injected via the tail vein into the male C57BL6 mice in order to examine whether the NRF1 mediates the inhibitory effect of microRNA 378 on hepatosteatosis. 8-week-old wild-type mice were maintained on the high-fat diet for 8 weeks. At 16 weeks of age, the mice were divided into 3 groups. A control group, a group injected with the microRNA 378 ASO, and another group that received a combination of the ASO and the NRF1 shRNA. The next step was the fatty acid treatment of HepG2 cells. And these cells were cultured in a DMEM medium, and they were cultured for 24 hours at first. And then the lipid accumulation and microRNA 378 expression was determined by an oil red staining. And then a fatty acid oxidation assay took place, and they were placed in XF24 cell culture microplates, and they were cultured overnight in a growth medium. The following day, the medium was replaced with a substrate-limited media, and the cells were incubated for an additional 24 hours. 1 hour before the assay, the cells were equilibrated with KHB buffer, and then incubated at 37 degrees Celsius for 1 hour without CO2. For the induction of FAO, palmitate BSA complex was injected at a final concentration of 100 micromoles, and the FAO capacity was represented as an increased oxygen consumption rate, OCR, in response to the palmitate BCA complex. And finally, a CHIP assay, or a chromatin immunoprecipitation assay, was constructed by the utilization of 130 milligrams of livers from the mice treated with either the high-fat or the standard diet. And then a binding region detected the PCR reactions, and the sequences of those were calculated. Anyways. So the results of the study show that microRNA 378 is robustly induced in fatty livers of obese mice and in human HepG2 cells exposed to high levels of fatty acid. So the researchers attempted to identify the microRNAs that are highly and specifically expressed in hepatocytes by comparing the microRNA profiles of livers of hepatocyte-specific dicer-1 knockout wild-type mice. Among the 88 microRNAs that were reduced after the dicer knockout, the microRNA 378 was reduced in threefold the dicer-1 knockout mice, suggesting that the hepatocytes represent a major source of microRNA 378 expression. The expression of the microRNA 378 is reduced in fatty livers of high-fat diet-treated mice and in the human HepG2 cells with accumulated lipid, leading us to focus on the microRNA 378. So with further examination of that, NRF1 is a direct target of microRNA 378. The potential effects of microRNA 378 on lipid homeostasis prompted the researchers to explore the downstream effectors of microRNA. To investigate whether the microRNA 378 suppresses NRF1 directly, they used a luciferase reporting construct in which the mutated microRNA binding site was embedded downstream of the luciferase, and the expression was represented by microRNA 378 and HEPA-1/6 cells, whereas its expression with the mutated sequence was not altered significantly, suggesting that a direct representation of microRNA 378 on the NRF1. Therefore, NRF1 is a direct target of microRNA 378. MicroRNA 378 also impairs FAO by modulating the expression of NRF1. NRF1 is a pivotal modulator of nuclear encoded mitochondrial proteins and mitochondrial biogenesis, suggesting that the microRNA 378 regulates FAO by targeting NRF1. In order to confirm this, the HEPA-1/6 cells incubated with the oleate were transvected with scramble NRF1, shRNA, and microRNA 378 ASO, and a combination of the two. The design allowed the researchers to determine whether NRF1 was required for microRNA 378 to impair FAO. As expected, the knockdown improved the FAO, and the additional knockdown of the increased NRF1 offset the effect of the microRNA ASO. The liver-specific expression of microRNA impairs FAO and promotes hepatosteatosis. MicroRNA 378 is able to induce hepatic lipid accumulation in mice. A microRNA 378 in vivo expression was generated using the mini-circle, the episomal DNA vector that's produced as a circular expression because that's devoid of any bacterial plasmid DNA. It was shown that there were no significant changes of the SREBP1C, SED1, FASN, and GPAT detected in the livers of mice treated with the MCTTR microRNA 378, which indicates that the impaired FAO rather than lipogenesis is a major contributor to hepatosteatosis promoted by the microRNA 378. As well, the NRF1 mediates the inhibitory effect of the microRNA 378 ASO on hepatosteatosis by modulating the NAFO. The mice that were maintained in the high-fat diet were randomly allocated into 3 groups, and with those 3 groups, it was shown that those inhibiting the microRNA 378 led to a slight decrease in liver to body weight ratio and a 51% reduction in hepatic triglycerides. The microRNA knockdown improved the ability of the hepatocytes to oxidize fatty acid, and the expression of the genes controlling FAO included CPT1, PPARC, and ACAD1. It was significantly increased in the microRNA 378 deficient mice. The findings from this particular experiment showed that NRF1 mediated the inhibitory effect of microRNA 378 ASO and hepatosteatosis via modulation of the FAO. The results also show that NRF1 inhibits the transcription of microRNA 378. The potential mechanisms were investigated by first predicting the potential binding sites for the transcription factors within the promoter regions of microRNA 378 using a Malin specter. The analysis revealed a highly conserved binding motif for the NRF1 within the promoter of microRNA 378, and the expression was reduced in fatty livers. The promoter of microRNA 378 was examined by the luciferase assay, and the overexpression of the NRF1 significantly repressed activity of microRNA 378 promoter. These particular findings showed that NRF1 overexpression strongly reduced levels of mature microRNA 378 and microRNA 378 primary transcript. And the chip sequence reveal that the DNA fragments containing NRF1 binding sites within the primary microRNA 378 promoter were immune precipitated from genomic DNA of mouse livers by an NRF1 antibody, indicating that the NRF1 can bind to the promoter region of the microRNA 378, and the NRF1 expression was lower in the livers of the high-fat diet-treated mice, showing that the binding affinity of NRF1 on the microRNA 378 promoter should be lower in the livers of high-fat diet versus the standard diet in mice. And finally, the liver-specific inactivation of NRF1 increases expression of microRNA 378 and promotes hepatosteatosis. NRF1 is a transcription repressor of microRNA 378, and microRNA 378 directly targets NRF1 through sequence. So as the beginning of the paper stated, it was hypothesized that a negative feedback loop between microRNA 378 and NRF1 maintained high levels of microRNA 378 and low levels of NRF1. This consequently promotes the proliferation of hepatosteatosis. And in order to test that specific hypothesis of the negative feedback loop, those 8-week-old mice were kept on high-fat diet for 8 weeks. And then this was the experiment that at 16 weeks of age, the mice were injected with the 3 different medias, one including the NRF1 shRNA to antagonize the endogenous microRNA 378 in the liver. Upon this, comparable increases of primary RNA 378 and mature microRNA 378 were observed, and this demonstrated the high-fat diet induced microRNA 378 as NRF1 dependent. So it was speculated from the researchers that the negative feedback loop could maintain high levels of microRNA 378 after NRF1 knockdown, which would enhance the inhibition of microRNA 378 and FAO. As expected, the NRF1 deficiency impaired the FAO, which indicated that microRNA 378 and NRF1 formed a negative feedback loop in fatty livers. Once this feedback loop was activated, microRNA 378 suppressed expression of NRF1 and reduced NRF1 further, and its suppression of microRNA 378 transcription. In consequence to this, the reduction in NRF1 impairs FAO, which leads to aggravated hepatosteatosis. Why are we glitching? I pulled up some of the charts from the paper. These graphs right here. The first one is the hepatic lipid accumulation, increased expression of microRNA 378, and the levels in the liver versus the wild type mice, and then the Dicer one. And then over here at graph B, the levels of microRNA in the livers of the high-fat diet mice. We have the high-fat diet mice right here, and the standard diet here. And this is the microRNA 378 levels. And then over here indicates the microRNA 378 levels comparison with the control medium without the oleate, and then the oleate as the control. And then here we have graphic representation of the microRNA binding motifs within the sequence. And this is a comparison between the mouse versus human DNA sequence and how they're complementary. And then over here, we've got the relative NRF1 levels in the standard versus the high-fat diet. And then this kind of moves along with the rest of the paper, luciferase activity, relative NRF1 levels of the control microRNA, and then also in comparison with the ASO, their microRNA levels. And here is The red oil staining of the three groups of the HEPA-1 6 cells treated with the scramble, the control right here, the ASO, the shRNA, and then the combination. I found these to be really helpful. It's easier for me to look at the graphs and understand the cell pictures. And then over here is the same concept. Moving down here, there are some other photos of the oil staining, the control, ASO, and then the combination. Continuing down here. And here is a good example of the loop between the microRNA 378 and the NRF1. So we'll start here, high-fat diet. High-fat diet induces microRNA 378's presence, and from there also activates the NRF1 over here, and then NRF1 proliferates fatty acid oxidation, FAO, and FAO aggravates and causes hepatosteatosis. I'm going to move back over here. And I just included this lovely chart of the image of a liver. This is the non-alcoholic fatty liver diseased liver, the histopathology, kind of the charts that we were looking at from the paper, NASH, fibrosis, and cirrhosis. And this kind of gives you the panel of what the histopathology looks like for each of those livers. I hope you enjoy this presentation, and I look forward to seeing your